Yeah, I hear you. Roger that. Rennie, would you mind zooming out there for me just so I can see where we're going? Yeah. Okay, great. Okay, great. I think science is still getting settled. Great. Do you want to come wide there a bit, please? I can do full wide for a minute there.
That's all we got.
Roger. Yeah, sure thing. How's the back row doing back there, guys? You guys all settled?
We're getting there.
Roger that.
Looks like it's all right.
Go ahead, Jonathan.
Got enough flashlights up here? What is happening? There's a nice umbellula right at the front of the porch. Sea pen.
It's a nice and blue.
It's waving at us.
Sure.
Sure.
Go for it.
So science, as you're getting settled back there, I hear we want a rock from this site here. So when you're ready, feel free to direct me to which rock you're interested in, and we'll try taking a grab.
Okay, sounds good. I think we're ready.
All set. Except, yeah,
So
the
I know
observers are just getting ready.
they tried for that crust and it broke.
Okay.
How about the rock to the right of the sea cucumber on the left-hand side? See the,
Oh, on the left-hand side.
to the left?
Left.
Yep. There's a rock about the right size, just to the right of the sea cucumber.
Is that telestrator working in front of you? This the screen right here. I'm pointing at it right there.
You have the tool.
This screen right here in front of you. You can poke
Where
that.
just
Right on the lasers there?
down.
If you poke at the screen, it'll drop a target on our screen up here.
Go to the,
There you go.
the, lower, the next one down, Paul. That rock.
Okay, great. Yeah, I'll reposition us and we can...
This one.
Oh.
Sorry, that
That
rock.
one. Okay, that one next to the holothurian to the right of it. Got it.
Okay.
Roger that. I'll hop over there and we'll grab
You
those
wanna-
rocks.
So you can.
You're all good there, Jake?
Yep.
All right, Jonathan, you want to do a quick zoom there, please? So is that the one you're looking for?
That looks like a good one.
Roger that. We'll see how large this one is.
Yeah.
Pull wide there, Jonathan.
Oh, don't have the coral cutters. Finally.
No coral cutters for us, no. I'm going to push in there a bit, please, Jonathan. Great.
Oh yeah,
Very thin.
that looks good.
Roger that. Go ahead and come a little wide there, Jonathan.
Can we get a good look at the rock before you drop it into the box?
Sure thing.
Look at your sheet. The sheet.
You want a little bit more?
Yeah, go ahead so they can see what's really on that rock.
So can we capture a still?
Oh, beautiful brittle star. And the tube is probably a spaghetti worm, the brown. Maybe we missed the worm, but we've got the tube. Maybe Kira will tell us if that's a phosphorite or not. But
Yeah, perfect. Let's get a still of the top of the rock if we can.
Sure.
okay, very good. I wonder if it's too big for the smaller boxes and might need to go in the forward port
Yeah, we can
space.
do whichever one you like. Yeah, it's getting to be a bigger rock, so we can do the toolbox. Science, what boxes do we have open back there?
We have forward on the port side.
Full wide there, Jonathan. Sorry, forward on the toolbox or forward on the starboard bio?
You call it the toolbox?
Yeah, the one that's right in front of us here.
Yep. We'll have to relabel our marker, our sheet. The starboard ones all seem to be small slots. Okay. So the forward one looks like the only big one.
Raj, oh, that's right. You guys do have your own boxes.
We have our own names.
Apologies for that, the confusion. All right, there. Jake, you want to go ahead and pop open the tool tray?
Yep, pumping it up.
Roger that. Any preference in which one?
I think they said
we only have one rock per box, so it has to be on the
port side.
Port side.
Roger.
Yep. Yep.
Jake, you want to follow me in on the bubble there, please?
Roger. Nice.
Oh, that's great. Thank you.
Can I have a sample number there?
All right,
Box
go ahead and
coming
tool
in.
there.
Science row.
Sample number?
Sample number is going to be NA124 014.
014. What was your number? 13. Let's double check because and the depth is 79.
All right, science, is there anything else you want to do around here, or?
Yeah, let's look for a second. Greg, what do you? Just trying to see if there's any of those white sponges we've seen before.
Yeah, do you want to put an arrow on that tool?
Just really quick,
zoom in on that guy.
I don't have a sample 013 in my log. Do you have a 013 in your log back there?
I think we're good for a zoom on sponge on the lasers.
Yes, please. We could slope those.
Is it a sponge?
Sure thing. So you want that one as a sample?
Yes, please, where the circle is.
Sounds great. Full wide there, Jonathan.
Full wide.
Raj.
So did we correct that discrepancy with the sample number?
Yeah, do we have an answer from the data row?
Was that last
We
rock?
have sample NA124 013 as a transect.
That is not a sample number. Unless it 16. Usually samples are physical samples, unless you guys have been doing different. Let's see, 6.
Yeah, Lisa considers the transect samples, so it sounds like they assigned it a sample number, but that may not jive with
Roger,
the
whatever.
normal numbering system.
Sure, whatever you guys are doing back there. So this is 014.
You want to angle Mongo up there a bit there, Jake, please?
Mongo up, yes.
Yeah, just maybe wrist up a little bit.
Let me just look at it with bubble.
Okay.
Wrist up and then wrist right to the bit.
That too much?
A little bit more, actually.
Better.
So I don't want to keep landing on it. Thanks.
Okay, so one number per transect. I guess they'll keep that labeling straight. 6, 7, 8. Yeah. I may be missing one then. Sample 9, 8 or 9. Yeah.
Where do we see our depth? Where do we see our depth?
All right, Jake, I'll have you ready on the slurp there.
Yep. Do we want to flush?
Yep, but first let's go ahead and change the cameras over.
Okay.
So you
Do
want
you want the sample salvo? We're coming?
We're
Or
going
no?
to slurp the
that's good.
sponges.
Look, it's on there. All right.
So go ahead and rotate to flush, please.
Flush.
Great. Stand by one, and I'll pull the hose out of the dirt.
Rudder.
Okay, go ahead, Jake.
Flushing.
All right, now I got our dust cloud in there. Okay, let's call that good, Jake.
Okay. What jar is this sponge going to go in?
We have coarse mesh and fine mesh, and this could go in a coarse mesh container.
Okay.
And so far we've only used one, and that was number 4.
Okay, the fine mesh are in 1, 2, and 3, right?
Yep.
Yeah.
Okay,
Any one of
so
those.
maybe jar 5. I believe that's 5.
That's 5, correct.
Yes.
You want to look at bubble cam on the sponge there, please?
Yep.
Great. All right, so
All right.
science confirming we're putting in 5?
Yes, please.
Great.
Yes.
Go ahead and turn on suction there, Jake.
Suction coming on.
Yeah, it looks like I have sample. I have one through 0.6, and then I have 2 transects, and then I have
Jonathan,
0,
push
1,
it on
0.
the HD bit there, please.
So I'm missing
That's good.
7, 8, or 9, one of those.
That's great on the sponge. There's another one just to the lower left
Okay, then.
of the laser, if that goes through.
Oh, this one down here?
Mm-hmm. Yeah.
Sure.
Thank you.
Of course.
Oh, there's that one.
Got
Yep,
it.
And
and
there's
one
another
more.
one. Okay.
So,
So that was in course 5, correct?
Okay.
Yep.
Yes.
Yes. Sample number NA124-015.
Okay, and then do we have then what is
Be
number
careful.
There
7?
it is.
0 at the suction.
0. 0 suction.
Oh, it looks like it was a snail. All right, Jonathan, come wide, please. Is that all you want in this sample jar there, Science?
Yes, thank you, Jessica.
Sure thing.
1, 2, 3, 4, 5. Okay, thank you. Just check on 1 through 7, if you don't mind. Thank you. And that was sample 015, correct?
Yes, confirmed.
Thank you. Don't even know what that was. What was it that we slurped?
It was a sponge and a
Sponge.
snail.
Okay, yeah, go ahead. Okay.
All right, Jake, when I lift off, I'm going to have you reposition
Uh-huh.
Mongo to get it out of the view.
Okay.
You can take the bubble cam on it.
Okay, so I'll just add 007 to that same area. I don't have enough space in the notes. It truncates it, so I'll just have this 1 through 7 all in the same spot. Got it. Thank you. Sorted out.
All right, Science, are we happy here, or did you guys want anything else around here?
I think we're good, please. We can keep going up the slope.
Roger that.
Roger that.
If we're heading
That's good.
directly to waypoint I,
Yes.
that should be, I'll bring, Argus will go 225.
225?
225. So
Roger that.
You're a bit,
225?
225.
325.
Roger. A 3,
225.
225.
225.
Roger.
Roger that.
You want that? Same.
Gonna go ahead and turn to 225,
Yep,
Roger.
bring in ahead to 225.
Rog.
So hi everybody, we've had a watch change and a new crew of science and pilots and navigators. And in the hot seat is Paul Jensen.
Let me know when you're ready then.
Go ahead, they're ready.
And
Bridge nav.
Yeah, hi everyone. My name is Paul Jensen, and I'm a microbiologist at the Scripps Institution of Oceanography. And we're interested in microbial diversity in the deep ocean. The microbes associated with the sediments that we've been collecting, and also with the various invertebrates that we see in these habitats, and also associated with these various hard substrates that we're seeing, especially the ones that are rich in various minerals like phosphorites and iron, manganese crusts. So to my right is Caitlin Creamer. You want to introduce yourself, Caitlin?
Sure. Hi everyone, I'm Caitlin Creamer. I'm a graduate student at Scripps Institution of Oceanography at University of California, San Diego. I am a student in Paul Jensen's lab, so I'm very interested in all of the microbes.
Can't hear you. Sorry.
All of the microbes in...
How's that?
Yeah. Better? I don't know if you can hear me or not. All of the microbes in the sediment, and what all the different processes that they're involved in, where they're living in different depths of the sediments, on and around the rocks, and as symbionts, some of these really cool creatures. So we're hoping to explore that.
Hi everybody, my name is Kyla Pearson and I am also a graduate student at Scripps Institution of Oceanography. I research deep sea worms
we got one.
in Dr. Gray Rouse's lab.
It's an anemone. And so we're going to continue on up the slope here. We're aiming for a depth. We're currently at 820 meters. We want to finish up at about 500 plus meters. And as we're going up, the oxygen will continue to decline. And we'll be seeking different kinds of rock substrates to sample from and other interesting animals that we can maybe take. We'll do a couple of different transects where we just will go along so we can do counts. And then we'll stop. We need to collect another 3 rocks in the next few hours. And also we'll be taking some slurp samples of various kinds of animals. What you can see now are quite a few sea cucumbers, the long white sausage creatures. They're called Panicia. We've got some large orange brittle stars, 2 kinds. Some soft corals are coming out on stalks. Those are called sea pens called umbellula. And lots of small animals are on the rocks, but we won't really see those until we can stop and zoom in.
Up to you. You can go ahead and try it off.
Oh, good idea.
No change. Guess the turbidity is not so bad.
Sorry?
I didn't see much of a change. Maybe the turbidity is not so bad.
Yeah.
Jess, we have 10 meters left on that move. Are you comfortable continuing up slope?
Yep, sounds great. Thanks, Rennie.
Bridge nav. Continue 50 meters, 225, speed 0.3. Thank you. We are...
can we just have a look at that white sponge that's right at the lower part of the frame right now?
Sure thing.
In fact, there's several ball-like sponges and a large sponge. I think it's going to be a glass sponge, Paul.
Covering all over that rock.
Jess, I'm slowing down to 0.3. They were at 0.5 before.
Roger that. All right, video, go ahead and zoom in. The big sponge there?
Yeah. Let's get a close-up to tell what kind of sponge it is. I think so. Are you interested in the other sponges? The lower ones? Can we pan a little lower, please?
Sure.
To the other sponges? Yeah.
Sure thing. There's
That's
a
the
few
same.
spot lobsters.
Yeah. We could slurp them.
Gonna stop the ship.
They'd come off easy.
Bridge now.
We could
They look
slurp over
similar.
the rock surface. We can just get a bit of a general slurp on this rock.
Hold position, please.
Sure thing. Go ahead and come wide there, Jonathan.
Or do you want to wait?
Let's wait.
Okay. He wants to keep going, so we'll keep going.
Oh, roger that.
Thank you. Sorry.
Oh, it's all good. I'll boogie out here a little in front of you there.
Roger.
Just to let you all know in the back row, so we're, if we stop to look at something, we have a little bit of time, as long as the ship's still moving. If so, just let me know if you want to prioritize being able to stop, also, or continuing on to cover ground.
Will do.
Bridge nav.
All right, Jonathan, you can go ahead and push past the core.
Let's continue that move, 50 meters, 225.
I don't know how to figure out here.
So just we're not moving directly upslope, it'll be slightly cross-cutting it in order to get to our steep bit.
Roger that. Thanks, Ernie.
Yep. Whoa, sub-bottom party. Yeah.
So maybe we can zoom in on this larger white sponge that's in the upper center of the field.
Sure thing.
Approaching.
Stand by there, Jonathan, please. All right, go ahead, Jonathan.
Yeah. So is that slurpable, or maybe that's too big, I think.
We can take a haircut of it.
Try the slurp, Greg?
Yeah, let's do it.
Sure
Bridge
thing.
nav.
And a full wide there, Jonathan, please. Thank you.
Let's hold position.
You could collect
Thank you.
the whole rock.
The whole rock might
Was-
be a bit large,
It's a big rock.
and it might limit us to collecting only very small rocks in the end.
Yeah, it's a 30, 40 centimeter rock.
Okay.
I do like collecting big things.
Well, Lisa, would that be a candidate for just being on the front porch and not in a bio box if we took the whole thing?
I don't know if the sponge would make it.
No. You should get your sponge.
Your sponge will
Okay.
not make it.
Okay.
I don't even think that would fit in the starboard front. I think we'll just slurp over this rock.
Roger that. All right, Jonathan, go ahead and push in there a bit, please. We'll get a full zoom on this guy real quick. Great.
There you go.
Getting some good stills there.
Yep.
Awesome.
You have a beautiful,
Hi, Crispin.
beautiful set of agglutinated forams living around the base of the sponge or on it. Pretty cool.
Science, would you like this in a coarse mesh or in a not coarse mesh?
Course for this
Course
one?
is fine.
Course. Roger.
Roger that.
So that would be jar number 6.
Yes, that can be jar number 6.
Beautiful.
We'll flush first, though.
And what type of sponge is this?
I think you're stretching our knowledge there, Kyla. Maybe someone on the chat can help us.
Sponge on a rock.
White massive sponge.
We're hoping it's a demo sponge.
All right, go ahead.
Flushing.
Take a flush.
Crispin, yes, the whale at Davidson Seamount that they collected on NA122 a few weeks ago, we had samples waiting for us on the ship. So yesterday during our down when we didn't have a day of diving, I was able to go through the samples and found some very interesting osidax and other polychaetes.
All right, let's call that
There.
good. Go ahead and 0.
That's cool.
That's great.
Oh,
I think you want
6.
6 there.
Yes, we're past it.
And do
There
you
it
want,
is.
would you mind looking at the sample with the bubble, please?
Roger.
All right, Jonathan, go ahead and push in there a bit, please.
Thank you.
That's great.
Data, can I confirm this is 016?
Yes, that is correct. NA124 016.
Ready for suction?
Roger that, thank you.
Yeah, so science, we're going to just take a bit of it, right?
Yes,
Not-
please. And around the base, there's the brownish stuff.
Roger that.
You can get some of that off too, would be good. Hopefully a piece will break off for you.
It's kind of like suctioning of
looks like-
Jello, isn't it? Get a little bit more down on this. Or flan.
Wow.
Flan.
Oh, interesting.
This is a flan sponge.
Got a little bit of a bite.
Got a piece?
Yeah, looks like
See
it.
what we got in the...
It's coming through now.
There's a chunk of something.
There's a piece of that sponge.
Give
See it
them a
swirling around the jar.
Okay, that's great. We could, oh, that'll be nice if you can
Nice.
get that other piece too.
Sure.
Hopefully it'll grow back.
Looks like we
Nice.
got a good sample here.
Great.
Looks great.
So which container did that go in?
It's in
Course
6.
Course number 6.
And the sample number?
NA124 016.
You can go ahead and 0 it there real quick, Jake, and see if
Roger.
anything falls.
Uh,
Sometimes it might get stuck on the side of the mesh too. Go ahead and cycle it one more time on.
yeah, firing it back on.
Full wide there, Jonathan, please.
Wiggle, wiggle, wiggle.
You know what they say about dancing with hoses?
I don't,
You
Jess.
can do the worm. Right?
Do the worm, do the worm.
Much easier to do the worm that way than. All right, well that's a bad joke, I guess.
Mark that one down under your tally.
It starts.
All right. Seems like most of it went in there.
You could do a little more on the rock, and we'll see what else we can come up with.
Sure thing. Go ahead.
Maybe a little squat lobster or other things that might be there. Not the anemone is probably not going to come off, but.
Roger that. Go ahead and push in there a bit there, please. So you want something else besides this half-eaten piece of flan?
Yes, please.
Roger.
The sponge is just for Paul. The other, any other things we can see what else there is.
Sure thing. We can try the squat lobster out here.
Now I'm hungry for flan.
50%.
Roger that.
There it goes.
So, Kyla, that's probably a Munida.
Okay.
Anything else on here that you guys might want? Go to 100%. Make sure they all get through there, please.
You might be able to get the anemone if you really want it.
Sure
You can
thing.
try and scrape it.
Yeah, we can definitely do that.
Up to the watch leads, though.
Make sure that we get our first two friends in there first.
They're still making their way through the suction.
Yeah, it's taking a bit of a time. Come a little wide there, please, Jonathan. Great. I'm going to circuit the
anemone be
sediment.
interesting?
No. Try the
Must be in the hose hanging on for dear life.
Science, I'm just going to get a little sediment so we get an indicator of where things are in the hose.
Fair enough.
Are you at 100% there, Jake?
Yep, 100%.
Also, the sediment came through. Oh, hey, Jonathan.
Doesn't want to come out.
Oh, there we go. We can see it move through the
Oh, nice. Yeah.
this side there.
There's a squat. Just went.
Oh, yeah.
Okay. More bits.
Is there cup coral in there? What are those little bits?
Looks kind of like a cup coral, huh?
So, Crispin, answering your question, we also had quite a few dorvalaids in the sample, some interesting amphirettids that they had noticed all around the whale fall at Davidson Seamount, and we picked up a couple of those.
Oh, more pieces.
The whole of the humerus is in the minus 80 and wasn't subsampled, so I'll be having to
Did you see
thaw
this?
that out to
I
subsample.
thought I saw the squat, but I couldn't.
Yeah, let me zero it, see if it's in there.
Later on, but
Good.
the samples I went through yesterday were from a
Yep, there it
vertebra
is.
Squat.
and
Got
a
it. Did
jaw.
you ever get
So,
that second chunk of the sponge?
so the slope is finally cleared,
I don't know.
and the squat
Good.
lobster and the rest of
Combine
the
with the other
sponge
one.
came through.
Maybe, yeah.
Uh,
All right. Did you guys want anything? You want that
Uh,
anemone, you said?
no, thanks. We'll pass on the anemone.
Roger that.
I think we'll index and call this one a sample. So we were waiting there while the crew were trying to get that slurp cleared. You can't see it in the feed, but the hose was a bit clogged with the squat lobster and a large piece of sponge, but with all of the jiggling they managed and a lot of flow, they got it through.
successful stop.
Yeah.
Yeah. All right, Jake, you can go ahead and line it back up on flush there. Thank you. All right, back on the road.
Yes, please.
Roger that.
Ready?
Yeah, go ahead, Trini.
Bridge nav.
Mind if I do a quick gauge check on bubble?
Please.
50 meters, 225.
Little red jelly.
As far as I know, is on deck at 4. I can. I think you're right then.
Not that. That one. So we've got a lot of pink sea urchins right in the frame here. This is Strongylus centrotus fragilis. They're very delicate sea urchins with fairly short spines and a fairly fragile shell, but they seem to tolerate low oxygen quite well. And otherwise, the long white sea cucumbers are called Panicia. And there's, we've been working a little on this group, and from the Eastern Pacific found that it's quite a few different species here in the Eastern Pacific margin that haven't been named yet. Pretty sure we've collected this one before, so we'll let these ones alone.
Just maybe for a little background too on why we're, the rationale behind what we're choosing to collect, and we tend to be focusing particularly on sponges because they have a history of producing interesting molecules that we call natural products, and part of this exploration is to understand what the potential is for the organisms that live in these deep sea sites to produce these types of molecules that may at some point have some value to us. So understanding these resources and what they may hold is an important part of this exploration. So organisms like sea urchins, I think you can immediately recognize that they have structural defenses, right? They have spines and shells, and if they're not going to be eaten, it's likely because they're structurally defended. So it's the things that tend to have soft bodies and no obvious structural defenses are the types of things that we're focusing on. And so we've already made a collection. The first collection we made was the sea cucumber that you see that's so abundant. So whether they're chemically defended and if that's the reason why they're not being eaten is one of the questions that we're interested in understanding.
Paul, did you collect some Panicia? These sea cucumbers?
We haven't collected one here, Lisa. We could. I have collected these before.
I just wondered if Paul wanted to look in them for.
Yeah, thank you, Lisa. We collected a sea cucumber already. It was a different one, though. You're right. So we haven't collected this one.
We could take one of these. I'm still trying to get a bigger collection for sequencing. Let's get to the next series of rocks, and I assume we'll find some there.
Are we at sort of a plateau now? Is that why there's so much sediment here? Are we not climbing, or is this still a slope? Can the pilots tell?
Sure, we're still going generally upslope. It has flattened out a bit here. It could be that the multibeam didn't resolve the, you know, it'll be steep at some parts, less steep at others, but generally trending upslope here.
There's also 11 more sediment cores, if you wanted to fill them.
Ooh,
we would.
Yeah,
That
this
means
could be
look at some.
This could be the time to do that.
Okay, would you like me to stop up here and we'll find a
Yeah,
spot?
yeah. That's
we could stop near a sea cucumber. Then we could get that as well.
Perfect.
Roger that.
I think we should probably also
hold position.
Take a water sample.
Yeah, before we settle on the bottom, let's trip a Niskin so we don't get any of the disturbed sediment into the water.
Roger that. Is there a preferred altitude that you have in mind?
I think where you are is fine.
Anywhere is fine. Roger that.
Thank you, Caitlin, for remembering that.
I'll just get out a little in front here.
Yeah, roger that. Might be a little bit of pendulum, but it shouldn't be too much.
All right.
Yeah, so our interest in sediments are from my lab's perspective, we're interested in the bacteria that live in these sediments because bacteria also are a source of these molecules we call natural products. And so we're interested in understanding if there are different types of bacteria that live in these sediments relative to the shallow water ones that we're more used to studying. And so we'll ask questions about the diversity of bacteria that live in these sediments, and then we'll also ask questions about their potential to produce these natural products, or that we're calling on this cruise their biopharmaceutical potential.
Full wide there, Jonathan, please.
And so we've already collected a number of cores which we can do this from, but now we're going to collect another set of cores from a different site, and this will give us plenty of material so that we can not only look at the bacteria that live in these samples, but Lisa is particularly interested in the small invertebrates that live in these sediments as well. So it provides an opportunity to assess their biodiversity as well.
So it's also
Gonna
important
look
to
at the Niskins there.
Yes.
Thank you.
Looks like 1 and 2 are triggered.
1 and 2. Yep. So we should be on Niskin number 3.
It's the blue string, I think.
Blue string.
Blue string, Raj.
This is going to be sample 017.
Roger that, 017.
Threaded or not.
Is there a color associated with that Niskin?
Blue.
Yeah, it's the blue one.
But it's going to be number 3.
Ready?
Watching.
Popped.
Fired.
Beautiful. Thank you.
Sure thing.
And about how far above the sediment was that tripped?
That was probably around 7 meters or
7
so.
meters, okay.
7 meters above sediment. Thank you.
Good point. Thanks, Jake.
to remember to take a note that the first one was tripped actually on the bottom after we disturbed the sediment a little bit.
Nice catch, Jess.
4. All right. And so now we're going to go for some sediment cores.
Yes, please.
Great.
So as far as placement goes, we're interested in maximizing the diversity of the microbes we see. So don't worry if you're putting them in places that look a little different. That's okay. We just don't want to put it on top of an urchin or another animal, an obvious animal.
Roger that. And what sediment cores have been triggered, or not triggered, but taken so far?
We have used all of the push cores on the starboard side.
Roger that.
So I think we'll be looking at using the core crate on the port side.
Great. And we're going to be going for 5 of them, or 4, or how many of you guys like here?
Lisa, are you listening? Do you want us to get all 6?
I think we might want to save some if we can get up higher.
Why don't you take 3 and save 3.
Okay, we'll do.
Beautiful.
Roger that.
Show you the manip.
Go ahead and show me that crate there, please.
Oh, okay. Coming back.
Came up with this hallway method.
And the hallway method's kind of cool.
Yeah. I dig it. I think we had those on OCNMS, right?
Yeah.
Man, these rubber bands, we're going to have to compromise them.
Industrially rated rubber bands. All right, so anywhere around here, sides?
Looks great.
Great. I'm just going to pull them one at a time if that's okay with you.
Sure.
Get it a little bit better. A bit stiffer than expected.
Yeah, it's possible there's rock underneath this layer of sediment.
Yeah.
So the good news is if we get 5 centimeters, we're happy.
Roger.
So we just don't know how much is going to survive. That's as deep as we can go. Is it? I don't know if you want to pull one out and just see how it looks, or if it's just going to fall out, then maybe this isn't a good spot.
Roger that.
Hey.
Oh, awesome.
Yeah,
You got that much. Would you guys?
That's a little marginal.
Yeah, we can also shake it out over here and try another spot, but it might be harder rock underneath.
Yeah, you could try another spot. And
Well, just so you know, for our lab to use the cores, they have to be at least 10 centimeters.
10 centimeters, okay, so.
But you can take as many as you want.
As maybe we'll try again, and if we don't get a very deep one, we'll just keep this one shallow one, but we won't take any more. We'll save the rest for hopefully a place where we can get deeper ones.
Sure thing. And I saw you circle a place.
Oh, I had actually accidentally touched the screen, trying to use the lasers to estimate if the lasers are 10 centimeters apart, estimate what the depth of that core was.
Roger, understood. I'll try one up here, but yeah, it might be the same type of business.
Roger that.
Oh, this one's looking a little more promising.
Hey, that looks good.
Push, push.
All right, so I think this is as happy as it's gonna go there.
That's quite a bit better, huh? So I'd say that's encouraging.
So we're gonna keep this guy here?
Yeah, let's keep it.
Roger that.
Looks good. Fantastic.
Lisa, that doesn't
Push core
look
number
like
11.
Jake, you wanna follow me in there, please?
Yes. Spin around.
Roger.
Well, that's a tough camera angle.
It's just my arm got moved around a little bit. Stand by. Sorry, guys.
It's going, it's going, it's going.
Yeah, I might lose it.
We don't have anything left in this guy.
Okay, it was a good try.
Yeah,
Sorry, guys.
Valiant effort.
There's no like clay plug under that's keeping it in.
Oh.
It's all that surface stuff.
Want me to try one more spot here?
Sure, give it another go.
I'm gonna reset my grip too when I'm down
A
there.
little sandier than when we were before, which is making it more difficult.
It's right here.
Yep.
That looks great.
Great.
Worse than before.
Yeah.
Even more shallow.
Not terrible.